Hello and welcome to Biology Interest. Today we're going to be looking at in vivo gene cloning as part of the A-Level Biology revision series. So in my previous video, we talked about creating a gene fragment. And now we're going to be looking at ways to increase the number of these fragments so there are in sufficient quantities to be used to create protein. Now there are two ways this can be achieved. Today we're going to be looking at in vivo gene cloning using a host cell. Now, using restriction endonucleases to create a DNA fragment, that's what we did in a previous video. We're now going to be looking at inserting these fragments into a host cell to help produce vast quantities of these fragments, and then eventually vast quantities of the protein we want. So now we've got this DNA fragment, we need to actually add extra lengths of DNA to it. For the transcription of any gene to take place, RNA polymerase needs to be able to attach to the DNA so it can go along it and create messenger RNA. And so for this we need a promoted gene. We need a promoted DNA sequence, a binding site almost, for the RNA polymerase to attach to it, to be told where to attach. And in a similar way, we also need a terminator sequence to be added to tell the RNA polymerase when to be released and to end transcription, when to release the messenger RNA it's created. And so we need to add those terminator and promoter regions to our DNA fragments we created. Once we've done that, we need to actually add our DNA fragment with the promoter and terminator genes to a vector, almost a carrying unit, something which is going to carry our DNA into a host cell. Now, a common vector is a plasmid, which is a circular ring of DNA often found in bacterial cells. Now, if the same restriction endonuclease is used on the plasmid and on the DNA fragment, we get the same sticky ends. This ensures both the DNA fragment and the plasmid, both the cut ends are complementary to the sticky ends, allowing it to join together more easily. We then use DNA ligase to connect these complementary DNA fragments and plasmids so it becomes a recombinant piece of DNA. It contains two pieces of DNA from different things. Once the DNA has been incorporated into the plasmid, we need to reintroduce this recombinant plasmid into the bacterial cells. This is a process called transformation. What you do in transformation is you have the plasmids and bacterial cells mixed together in a medium containing calcium ions. We then rapidly heat it in a process called heat shocking. This makes the bacterial membrane more permeable, allowing the plasmids to pass across the membrane into the bacterial cell. However, when we do this, only a few bacterial cells actually successfully take up the recombinant plasmid. The bacterial cells also take up a variety of different DNA fragments that have formed loops or plasmids that have reformed because they all have the same complementary ends, which is the problem when you use the same restriction enzyme to cut them. So only a few of them actually contain the recombinant plasmid that we actually want. So we need to actually test and be able to tell which cells have taken up the recombinant plasmid successfully. One of the ways we could do this is by using antibiotic resistance. So let's say the plasmid has two antibiotic resistance genes. Now, what you would do is you'd grow the bacterial cell in a growth medium containing one of the antibiotics. And this means that only the bacterial cells which have taken up the plasmids, as only those contain the antibiotic resistance genes, they're the only ones that survive. The bacterial cells which just take up the DNA fragments which have rejoined together die because they don't contain the antibiotic resistance gene. However, some of the cells which have taken up the plasmids and the recombinant plasmids have taken up only plasmids which don't contain the recombinant DNA we're looking for. So we now need to distinguish between the recombinant plasmids alone and the plasmids which are just by themselves. What we want is the recombinant plasmids. And so to do this, we have to look at another antibiotic resistance gene. And so what you do is you have this gene cut. When you cut the plasmids, you cut the plasmid where the second antibiotic resistance gene is. This means in the recombinant DNA, this antibiotic resistance gene doesn't work because it's been cut and split and the new DNA fragment has been inserted. So that gene doesn't work for antibiotic resistance. This means you place samples of the bacteria that has survived the first antibiotic onto the new antibiotic, and the ones that die are the ones that contain the recombinant DNA, because you've broken the antibiotic resistance gene when you cut it with the restriction enzyme. The ones that survive are the ones that contain just the plasmid, because you haven't cut that, and so you can discard those because you don't want them. So that's how you successfully worked out which DNA out of the DNA you've created, out of the plasmids you created, are actually recombinant plasmids and the ones you want. Now, apart from using antibiotic resistance to help show you this, you can also use fluorescence, so you can have a gene which fluoresces, which tells you which thing is the recombinant DNA, or you can use an enzyme which produces a particular enzyme which tells you which bacterial cell is recombinant. So I hope you enjoyed watching this video about in vivo gene cloning. Stay tuned for more videos from biology interest. Thank you.